What do we really mean by specifications and analytical methods in pharmaceuticals? These concepts are at the core of drug quality, safety, and consistency. Every medicine that reaches a patient must meet clearly defined standards, and those standards are ensured through testing. Let us start with specifications. Specifications are a set of predefined criteria that a drug substance or drug product must meet to be considered acceptable for use. They act as a quality benchmark. If a product meets all specifications, it is approved. If it fails even one, it is rejected. Specifications are not random. They are scientifically justified and based on development data, regulatory guidelines, and manufacturing experience. They ensure that every batch of medicine is consistent in quality and performance. Now let us understand analytical methods. Analytical methods are the procedures used to test whether a product meets its specifications. These methods are developed using scientific techniques and instruments such as chromatography, spectroscopy, and titration. In simple terms, specifications define what is required, and analytical methods determine whether those requirements are met. Next, let us talk about tests. Tests are individual checks performed on a drug substance or drug product. Each test evaluates a specific quality attribute. Some common tests include identification, which confirms that the correct drug is present. Assay determines the amount of active ingredient in the product. Impurity testing checks for unwanted substances that may be present due to synthesis or degradation. Dissolution testing evaluates how quickly the drug releases in the body. Microbial testing ensures that the product is free from harmful microorganisms. Each of these tests plays a critical role in ensuring the safety and effectiveness of the medicine. Now let us move to acceptance criteria. Acceptance criteria are the predefined limits or ranges within which a test result must fall. They define what is considered acceptable. For example, the assay of a drug product might be required to be between 95% and 105% of the labeled amount. If the result falls within this range, the product passes the test. If it falls outside, the product fails. Acceptance criteria are established based on scientific data, including stability studies, clinical data, and manufacturing capability. They are designed to ensure that minor variations do not affect the performance of the drug while still maintaining strict quality control. Together, tests and acceptance criteria form the backbone of specifications. Now let us understand validation basics. Validation is the process of proving that an analytical method is suitable for its intended purpose. It ensures that the method produces reliable and consistent results. There are several key parameters involved in method validation. Accuracy refers to how close the measured value is to the true value. Precision measures the reproducibility of results when the test is repeated. Specificity ensures that the method can accurately measure the desired analyte without interference from other substances. Linearity evaluates whether the method provides results that are directly proportional to the concentration of the analyte. Range defines the interval within which the method is accurate, precise, and linear. Robustness assesses how small changes in method conditions affect the results. All these parameters are evaluated during method validation to ensure that the method is dependable. Validation is not just a one-time activity. Methods may need to be revalidated if there are changes in the process, equipment, or formulation. Understanding specifications and analytical methods is critical in pharmaceutical development and manufacturing. They ensure that every product released to the market meets the required standards of quality, safety, and efficacy. Regulatory authorities carefully review these aspects before approving any drug. Manufacturers must demonstrate that their specifications are appropriate and that their analytical methods are validated. In conclusion, specifications define the quality requirements, analytical methods provide the means to test those requirements, tests evaluate specific attributes, acceptance criteria set the limits, and validation ensures reliability. Together, these elements form a robust quality control system that protects patients and ensures trust in medicines. Every tablet, capsule, or injection you use has gone through this rigorous process, ensuring that it is safe, effective, and consistent. This is how science ensures quality in every dose.
Subscribe to MedPharma Global on YouTube. Get the latest medical and pharmaceutical updates. Click the subscribe button and join our community.